Hello, everyone. I'd like to welcome you to the Phytobiomes Alliance webinar series. Today, we'll be talking and learning about engineering nitrogen-fixing microbial communities associated with maize and sorghum roots. And I'm pleased to be joined today by Jean-Michel Anet, who will be giving our webinar. But before I do that, let me give you a little bit of a background on the Phytobiomes Alliance. So we are a nonprofit consortium of industry, academic, and governmental scientists. And I want to thank our sponsors who make this possible for us, both the webinar and all of our activities. And to remind you that the phytobiome is really the complex system of plant-based agriculture, or doesn't even have to be agriculture, but could be a forest. But it is the geophysical and the biological components that interact with plants as they are growing in a substrate, whether that's soil or in a container in a controlled environment. And it's all the microbes, the other animals, climate, weather, water, but it's also influenced by management practices, and we need to understand how those management practices actually interact with that entire system. So as I mentioned, phytobiome is everything from crops to pasture to forest, vertical farming, vegetable gardens, etc. Our vision is that all farmers will have the ability to use predictive and prescriptive analytics based on the geophysical and biological conditions to be able to really determine what will be the best combination of crops, in fact, what variety in some cases, what management practices and inputs they should use for their particular site in any given year. We do have a couple of upcoming Phytobiomes Alliance events. We have a genome sequencing and microbial resources workshop that's being put on by the U.S. Culture Collection Network. We also will have another webinar presented in September from Prasanna Konkanala from Trace Genomics. And then we'll have our conference in October, Harvesting the Future, Bridging Phytobiomes, Agriculture and Climate. And I hope that you'll be able to go. We're calling for abstracts now, so we really would like to see a diverse group of individuals who participate in our conference, and it will be in St. Louis. So just to let you know, the webinar is being recorded and will be posted on the Phytobiomes Alliance YouTube channels later today. The presentation section will be followed by a Q&A period, and you can submit your questions in the Q&A panel. You can use the chat panel to contact and see messages from the organizers. And you should already have in the chat handouts of both this particular presentation about the Phytobiomes Alliance, as well as Jean-Michel Anet's presentation. If you want to keep up, be sure and register to the mailing list. So at this point, I want to turn it over to Jean-Michel Anet. He is from the Department of Plant and Agro Ecosystem Sciences at the University of Wisconsin and is well known as someone who understands and is promoting engineering nitrogen-fixing microbial communities generally, but today he's going to talk about maize and sorghum roots specifically. So thank you for being with us today, Jean-Michel, and I will turn it over to you.
Great. So thank you very much for giving me the opportunity to talk about our work on engineering nitrogen-fixing bacteria and microbial communities. And today I'm going to talk in particular about those associated with maize and sorghum roots. So I'm going to start the presentation by reminding you of the big picture of what I'm going to talk about, the nitrogen cycle. Just to remind you that most plants, most crops that we grow take their nitrogen from the soil, mostly in the form of nitrate and ammonium. What we call nitrogen fixation is the conversion of atmospheric nitrogen into forms that the plant can use, and it can be done in different ways. industrial fixation is also called the Haber-Bosch process that was invented at the beginning of the 20th century, is currently the way we produce synthetic nitrogen fertilizers. And as I'm sure you know, we currently use a large amount of these fertilizers, in particular for cereal crops. You also have atmospheric fixation that happens when you have lightning, but today I'm going to focus on biological nitrogen fixation, which is performed by bacteria in archaea that process and express the nitrogenase enzyme. As I'm sure you also know, some of our crops, like legumes in particular, associate well with some of these nitrogen-fixing bacteria. Legumes associate with rhizobia bacteria, and that's why these legume crops require little to no nitrogen inputs. That's why also they can even add nitrogen to the cropping system, in particular through crop rotations. Unfortunately, cereal crops like maize and sorghum, at least the cultivars we currently use, associate fairly inefficiently with nitrogen-fixing bacteria. So to put numbers on the different types of nitrogen fixation that I just mentioned, atmospheric fixation, as you can see, it's relatively minor, less than 10 million tons per year is fixed. Biological nitrogen fixation on land, so excluding what happens in the oceans, estimates vary between 90 to 40 million tons per year. And the industrial nitrogen fixation process, according to recent numbers, now exceeds biological nitrogen fixation, and it's about, or probably even above, 170 million tons of nitrogen per year fixed by that process, which is actually, again, fairly recent in our history. The Haber-Bosch process, right away, you can see it's going to likely cause a major disruption of the nitrogen cycle, given the amount of nitrogen that we now put on land. But first, I want to say that the Haber-Bosch process, or synthetic nitrogen fertilizers, are extremely useful and absolutely necessary for our current cropping systems. So here it's a simplified graph from that paper published in Nature Geosciences, just simply representing the growth of the world population in red from the beginning of the 20th century to now, And here in blue, the blue line represents the fraction of that population which is currently sustained, fed through synthetic fertilizers. And today we estimate that about half of the world population is sustained through the use of these synthetic fertilizers. So really nobody at this point thinks it would be reasonable to not to use synthetic fertilizers. So they're absolutely necessary. These synthetic fertilizers are, again, the Haber-Bosch process uses natural gas as a source of energy. And we use, I think, 1 or 2% of the world energy to produce these synthetic fertilizers. So we use a lot of natural gas to produce synthetic fertilizers to grow our crops for food, fiber, and biofuel production. When you think about what I was saying just before, that we feed half of the world with synthetic fertilizers, what that really means in practice is that we feed half of the world on natural gas, a non-renewable resource. And so I think that should be concerning for the long-term sustainability of how we feed the world. Another long-term concern or systemic concern is the fact that we now use natural gas to produce fertilizers, to produce biofuels. So obviously there is a obvious inefficiency in the system. Doesn't make sense to use natural gas to produce biofuel. These are long-term concerns, but I'm sure that you have heard that there are some more pressing and more short-term concerns with our strong dependence on synthetic fertilizers. First, for developing countries, both the cost of these fertilizers, but also the logistics of getting that fertilizers to smallholders is an issue. I want to highlight that book from Roger Furrow that I really loved, that both actually shows both issues for corn growers in smallholders that grow corn in Kenya. In many places where fertilizers are used extensively, the problem are environmental. And I'm sure that you have heard about nitrogen leaching and the issue of eutrophication of streams, river, and the degradation of coastal zones, where the case of the Gulf of Mexico, where we have eutrophication in the Gulf of Mexico, which is a direct consequence of the fertilizers we use in the Midwest. You also have an issue regarding the fact that these fertilizers are degraded into greenhouse gases like nitrous oxide that also contribute significantly to global warming. So for all these reasons, and in fact, for quite a long time, the community realized that we should rely not only on synthetic fertilizers, but also maybe even more, a bit more on biological nitrogen fixation. So that biological fixation, again, is performed by bacteria in archaea that process that nitrogenase enzyme. And there has been a lot of research on the mechanisms of that enzyme with both the dinitrogenase and the dinitrogenase reductase. I'm showing you this figure just to remind you 3 issues that we need to tackle when we want to use biological nitrogen fixation efficiently. One is the fact that that chemical reaction of breaking the triple bonds of dinitrogen into ammonium requires a lot of energy, whether you do industrial fixation or biological fixation. But in biological systems, the systems use ATP, and the bacteria that perform fixation will need a lot of ATP, and so an abundant source of energy to produce all that ATP. And for bacteria associated with plants, that energy usually comes into the form of carbon sources, photosynthase coming from the plant photosynthesis. The second issue is that these proteins have iron-based cofactors that can be oxidized really easily, and the nitrogenase is highly sensitive to oxygen. And so in any efficient nitrogen fixation system where you have biological nitrogen fixation, either the bacteria and sometimes the plant need to help to protect that nitrogenase against oxygen. And many of our what we call diazotrophs bacteria often prefer low oxygen environments. Some of them, like Azotobacter, can function very well in totally aerobic conditions, but many of them are facultative anaerobes that prefer low oxygen environments. And the last aspect is the transfer of nitrogen from the bacteria that fix nitrogen to plants. These diazotrophs fix nitrogen primarily for themselves, and they have actually mechanisms to conserve that nitrogen they just fixed. And so very often that transfer of fixed nitrogen from the bacteria to the plant is limiting, in particular if you're talking about bacteria that live in the soil, sometimes away from the plant. And so when we look at the history of my field of research on biological nitrogen fixation, when it comes to non-legumes like cereals in particular, people have taken approaches that are more on the plant side, and some approaches are more on the microbial side. Historically, the first approach that was pursued and was the idea of taking the legume system, these root nodules formed by rhizosphere on legumes, and try to transfer that to non-legumes. It's clearly not a novel idea. Here I'm showing you a bulletin from the University of Illinois from 1917, where the goal of that book was actually to see if symbiosis possible between legume bacteria, rhizosphere, and non-legume plants. So that idea has been in the mind of many decades of researchers, and progress has been limited. I think that approach is still very interesting, very promising, especially since for the last 20 years, where we had a better understanding of the plant genes in particular, controlling the formation of root nodules and infection by rhizosphere, and also the evolution of that symbiosis. So I think it's a promising approach, but it's clearly long-term. Along these lines of promising but long-term approaches is the idea of taking the nitrogenase enzyme and putting it directly into plants. So here it's an approach which is totally plant synthetic biology, and that several colleagues like Luis Rubio or Chris Voigt, for instance, are working on trying to express that nitrogenase into either plant mitochondria or plant chloroplasts. Both ideas I think are really interesting, worth pursuing, but they are long-term. And so I was interested in my lab in trying to see, can we also develop approaches that are more shorter-term to deliver more solutions in the shorter term. One of them is to explore host diversity. And can we find cereal crops or plants that are accessions of our cereal crops like maize and sorghum that would already naturally associate better with some of these nitrogen-fixing bacteria. And I don't know if I've heard about some of my projects on looking at fixation that happens in particular in what we call the aerial roots produced by maize and sorghum. So there are some accessions of maize and sorghum that produce roots, nodal roots on the stem. And when it rains, these roots produce a gel, a mucilage, which is a really good environment for nitrogen-fixing bacteria. And we show that these maize landraces get over 50% of their nitrogen from the atmosphere. And we have data that we are currently trying to publish showing that the sorghum accessions get about 40% of nitrogen from the atmosphere. So there are things that work already in nature in maize and sorghum. But these accessions are fairly different. And to put these straight into our current cultivars requires breeding, and it takes time. So the approaches I want to talk about today are focused on the bacterial side. Just the idea of taking naturally occurring diazotrophs on the surface of maize and sorghum and trying to modify them to increase their nitrogen fixation rate and force them to release that nitrogen. It's basically the idea of what we had here in that cover that we published in ACS Synthetic Biology, where we had our Willy Wonka nitrogen factory with these little nitrogen fixing bacteria performing nitrogen fixation in the vicinity of cereal roots. So that's what I'm going to talk about today. So first, I need to say that this approach is based on decades of research on the nitrogenase enzyme and on mechanisms controlling the expression and regulation of the nitrogenase enzyme. So the genes controlling the synthesis of the nitrogenase, the structural genes are NIF, H, D, and K. And then you have many genes that are necessary, additional what we call NIF genes that are necessary for the assembly of the nitrogenase. You also have regulatory genes like NIF-A in particular in proteobacteria, and in gamma proteobacteria, we have NIF-L, which is a negative regulator of NIF-A. And NIF-L and NIF-A are within the same operon. So I'm going to talk quite a bit about NIF-L, again, negative regulator of NIF-A, and NIF-A, a positive regulator of NIF gene expression. So I'm sorry for that graph, which is a bit crowded, but I'm going to go through it and to explain you what we know about the regulation of the nitrogenase and how we use that to engineer what we call ammonium excreting bacteria. So GLAN-D is a protein that perceives the nitrogen status of the bacteria. And GLAN-D has a UTase activity that uridylates P2 proteins. These P2 proteins control the phosphorylation of NTRB and TRC. And NTRC is a master regulator of genes involved in nitrogen metabolism in many bacteria. In the case of nitrogen-fixing bacteria, and in particular in gamma proteobacteria, uridylated P2 proteins are going to activate NIF-A by basically removing the repression of NIF-A by NIF-L. And so again, NIF-L is a negative regulator of NIF gene expression, and NIF-A is a positive regulator of NIF gene expression. And so a classical approach that I'm going to talk about more that we use to increase the nitrogenase activity in bacteria is to remove NIF-L and activate NIF-A. These P2 proteins also regulate the activity of GLAN-E, and GLAN-E itself is controlling the activity of the glutamine synthase. And so you have a set of approaches that aim at reducing the glutamine synthase, the GS-GOGAT activity. And it can be done by playing with GLAN-E, and in particular by, for instance, boosting the ATase activity of GLAN-E. It can also be done by affecting the promoter in front of the glutamine synthase, or even the glutamine synthase itself. But the goal here, the idea, is that we can promote the buildup of ammonium inside of these diazotrophs by decreasing the glutamine synthase activity. So again, strategy number one, we increase nitrogenase expression. Strategy number two, we decrease the assimilation of that ammonium by the cells. Oh, sorry. And the third point, which again is regulated again by these P2 proteins, are the ammonium importers. Many nitrogen-fixing bacteria do everything they can to conserve the nitrogen they just fixed. Again, it's very expensive for them, so in terms of ATP, so they have these AMTB, ammonium importers. And for us, if we want that ammonium that, again, builds up inside of the cell to leak out of the cell and to be released to the plant, we often delete these AMTB importers. So these are basically the three categories of targets that we are working on to make ammonium excreters, Nife, in particular in gamma protobacteria, everything about glutamate synthase activity and the MTB transporters. So to show you that it works, and here I'm citing, I'm showing a paper was a collaboration with the Pivot Bio company. It's a paper on, it was a paper on using Klebsiella varicola. And again, it's a gamma protobacterium and it has Nife. And so the idea here in that paper was to replace Nifel by IMC, which is an actually endogenous promoter of Klebsiella, a strong promoter, to replace, to delete Nifel. And Nifel, again, is right in front of Nife. So in just one manipulation, very simple manipulation, we can remove the negative regulator and activate the positive regulator. And we, of course, need as a control, they actually made a mutant in Nifh that do not fix nitrogen. And so under no nitrogen in the environment, the system works exactly as expected. That's the here nitrogenase activity measured by acetylene reduction assay in culture of the wild-type Klebsiella. When you have a delta Nifel IMC, when you put that IMC promoter in Nifel, you have an increase in nitrogenase activity and, of course, no activity at all when in the delta Nifh mutant. What's interesting is that even in the presence of ammonium in the medium, 5 millimolar ammonium, you still see a fairly reasonable nitrogenase activity in the delta Nifel IMC, which is interesting because it means that even in agricultural fields where you have nitrogen, you still get a decent nitrogenase activity. It works in culture. It also works in plants. So that was done in my lab using these very simple assays in pouches where we do what we call in planta ARA, acetylene reduction assay. You still see a good amount of nitrogenase activity when these engineered strains, delta Nifel IMC, are on plants. And of course, we're interested in figuring out the nitrogen release. And we see that these delta Nifel IMC mutants of Klebsiella release about, 1.5 millimolar ammonium in the medium, which is pretty good. We can play that game quite a bit with the different targets that I talked about. And here I'm showing you results from another paper where actually, again, with Pivot Bio engineered a range of ammonium excreter. Here it was in Cosaconia, but that's the same target, same ideas. And here that's the wild type strain. And here that's a range of strains producing, you see, with varying levels of nitrogenase activity. And what you see is that if you're looking at the growth rate of these bacteria, you have a clear decrease in growth rate, meaning that the more you engineer these bacteria to fix nitrogen, to have higher nitrogenase activity, the more their fitness is affected. And that's really what I want to tackle today and talk about today is that fitness decrease. So here, that was an example in Cosaconia. We see the same in Klebsiella. I wanted to show you another paper that we published with John Peters and Florence Mus, and on a third one, which is Azotobacter vinelandii. So Azotobacter is a fantastic nitrogen fixer. It's a strict aerobic bacterium that has its own way of protecting its nitrogenase through respiratory protection. And so we thought, great, it's going to be a great model to make that kind of manipulations. And so that's what we did with Florence and John, and to put that KICS cassette that contains that strong promoter inside of Nifel. So again, disrupt Nifel, activate Nife. And interestingly, these two strains, FM4 and FM5, these modifications were lethal to the bacteria. So the bacteria literally were dying. And so we actually demonstrated in that paper that in FM4 and FM5, if we mutate NIFH or NIFD in Azotobacter, you actually can restore the lethality effect. In other words, that modification in Azotobacter kills the bacteria from fixing too much nitrogen and probably consuming too much ATP. So that's the extreme case of fitness defect. It literally kills them. I wanted to show that paper also as a way to show that we still have to learn quite a bit about nitrogen fixation. Even if we think we know a lot about the regulation of the nitrogenase, we had actually an interesting surprise in that paper, because we inserted that KICS cassette also in reverse orientation with the idea initially of just knocking out Nifel without affecting Nife. And what we saw is that in these FM1, FM2, or FM3 mutations, we are seeing actually a very good ammonium excretion. And you see, after 48 hours, we reached 10 millimolar ammonium in the medium. And there we realized that, oh, but these mutations not only knock out NIFEL, they also activate RNF1 on the other side of NIFEL in Azotobacter. And we confirmed that by just taking just this promoter, just the endogenous promoter of APH, and using these promoters to, again, disrupt NIFEL and activate RNF1. And again, same phenotype, we see that we can reach really good ammonium excretion by knocking out NIFEL and activating RNF1. We, by the way, also patented that trick with John Peters and Florence. We showed that you can do that with different promoters, and we use these different promoters that are from Azotobacter that we were just moving around inside of Azotobacter. And so we use that APH promoter or SID-AB or SIG-B promoter. And we saw that the first two promoters actually gave us much more ammonium excretion than the third one. And that was surprising for us because these are the, that graph on the right, panel C, shows you the activity of these promoters. In other words, the strongest promoter, SIG-B, here in yellow, is actually not the best one for ammonium excretion. And so that shows really the limit and the complexity of developing these ammonium excreters. It's that it's a balance. We want to avoid that. We need to have that balance between ammonium excretion and fitness, but also here, we don't think it's about lethality. We need to have the right expression level of RNF1 with these first two promoters to maximize ammonium excretion. And so it's really a game where we need to balance a lot of parameters. So what I want to talk about today for the rest of my talk is what are we doing to try to find that balance and improve the balance so that it's beneficial for us in agriculture to use these ammonium excreters. So for instance, what several companies are doing currently is make a lot of ammonium excreters and find that optimum between ammonium release and the fitness decrease. With the idea, again, that the more these bacteria are going to fix nitrogen and release ammonium, the more that their fitness is going to decrease. I can say that from personal experience, when we try to find that optimum in the lab or in the greenhouse, it does not translate to the field. And so one message from my talk is that if you want to test ammonium excreters of nitrogen-fixing bacteria, don't rely too much on lab experiments or greenhouse experiments. Go to the field as quickly as possible, because that's the only test, the best test to find that balance, to find between, again, fitness and ammonium production. So today I want to talk about, with the time I have left, I want to talk about 4 approaches that we're working on in my lab. It's still work in progress for most of them, things that we are trying to find that better balance and have maximize ammonium excretion while maintaining good fitness. One approach is based on synthetic biology, is basically to control when and where these bacteria fix nitrogen. The strategies I showed you before are constitutive. We were just putting strong promoters inside of NIFEL or other places to trigger ammonium release. Let's do that now in a more controlled manner. That's the synthetic biology approach. The second one is trying to understand better how our ammonium excreters and diazotrophs interact with other bacteria in the rhizosphere. And we tackle that using synthetic communities. And in particular, from that work, we want to identify both the competitors of our diazotrophs, but also what are potential helpers. And so I'm going to talk about a third approach, which is how can we find more of these helpers, bacteria that help our weak engineered diazotrophs. And the last aspect is a bit similar, finding helpers, but getting some help from our little fungal friends, the arbuscular mycorrhizal fungi. So first approach, synthetic biology. Here, the idea is to not to work with ammonium excreters where we put constitutive strong promoter to disrupt genes, and to try to develop bacteria that fix nitrogen and at high rates and release nitrogen only in the presence of the plant when it's really necessary, with that these bacteria don't have to exhaust themselves releasing ammonium when it's just not necessary for the plant. And so the idea is, can we have a system where we take a plant that would produce signals, signaling molecules. They can be flavonoids, arabinose, or many other molecules from the rhizosphere to be perceived by biosensors that would then trigger the ammonium higher nitrogenase activity and ammonium release. So that idea was the basis of a paper which was led by Chris Voigt's lab. My lab was participated in that study, and where the approach was basically a lot of what Chris Voigt is doing, which is refactoring the nitrogenase complex, which is a great idea to get control, and I would say almost absolute control over the expression of the NIF genes. That refactoring process allows, again, a great control and allows controlling the expression of NIF genes with biosensors. And in that paper, we tested a wide range of biosensors for salicylic acid, nopalin, octopine. And we were able to show that we can get inducible nitrogen fixation in bacteria that actually don't even fix nitrogen originally, like E. coli or Pseudomonas protegens PF5, which is a classical plant growth promoting bacterium, or here that Rhizobium RBG74 or Rhizobium colinodons. So in that paper, using a lot of synthetic biology and really a refactoring of the NIF genes, we were able to show that, yep, you can get a great control of nitrogenase activity. Unfortunately, if you look at the levels of nitrogenase activity achieved in that paper, they are very low. Because by refactoring all these NIF clusters, we also lose a lot of the activity, and we were not able to show any ammonium excretion. So here we were able to achieve inducible fixation of nitrogenase activity, but not inducible ammonium excretion. A bit of the same idea was published by the group of Philpool and Giles Oldroyd were involved, and Tim Haskett was the first author on that fantastic publication in PNAS, where they showed that they were able to produce molecules called rhizopines into barley or even Medicago truncatula roots, and they used the rhizopine biosensor. They made a biosensor to drive the expression of NIF-A. And so the idea here is what I said before, is a plant signal inducing here NIF-A expression. And they did that in Rhizobium colinodons. And they were able to show, these are the data here, that again, they are able to achieve really good inducible nitrogenase activity. Unfortunately, Rhizobium colinodons is not a great fixer in terms of rate of nitrogen fixation, and they were not able to show inducible ammonium release. I need to highlight that what I think beautiful in that paper is the fact that it's a fairly orthogonal system, because they have a molecule which is not naturally produced by barley or Medicago that induces the system. So it's a fantastic paper. To go a bit beyond that and to achieve what that not only inducible nitrogen fixation, but also inducible ammonium release, we wanted to take a step back and figure out what is the best chassis for us to play that game of synthetic biology with associative diazotrophs. So last year we published a paper in collaboration with Brian Flegger, and Maya Venkataraman is the first author on that paper, and where we evaluated the different possible chassis for synthetic biology on diazotrophs. We worked on Klebsiella varicola that my lab isolated from sorghum. We use Pseudomonas studzerii, and again, that fantastic model Azotobacter vinelandii DJ that has been in the community for a while. And we evaluated how different biosensors, some of them, by the way, were coming from Chris Voigt's lab, were functioning in these different backgrounds. And long story short, clearly for us, Klebsiella varicola stands out as the best system to do synthetic biology in diazotrophs. Again, that strain that we call A3 was isolated in my lab from sorghum. It's Klebsiella, so it's a gamma proteobacterium, really close to E. coli. It's genetically tractable. All our biosensors pretty much work in Klebsiella. And most importantly, it's a great nitrogen fixer, and it's a great maize and sorghum endophyte, and epiphyte and endophyte. So it's a fantastic platform. So what we did, and what Valentina Infante in my lab did first, was to evaluate these different biosensors on plants and figure out which ones can respond to molecules from root exudates. And we found just, for instance, a few sensors that worked really well: arabinose, salicylic acid, and vanillic acid biosensors. Here, that's the response with just water. And here, these are good exudates of different cereals that's showing that we can get really good induction using these biosensors in Klebsiella varicola in that nitrogen fixer. So I won't have time to show you, but we're working also with other new types of biosensors, and we're also trying to improve the sensitivity of some of these biosensors in Klebsiella varicola. So at least these three work right away. To show you that the system works is that first we wanted to achieve, can we get what we had before, which is inducible nitrogen fixation. And again, the trick is fairly simple. We take these, here I'm showing you the example for the arabinose biosensor. We took that arabinose biosensor, we use it to replace NiFeL and to drive the expression of NiFeL. And it's working. Here it's the wild type, the nitrogenase activity of the wild type Klebsiella strain. Here that's the delta NiFeL, in which the mutant in NiFeL, in which we put the arabinose biosensor. No activity in the absence of arabinose, good activity in the presence of 0.02% arabinose. And we were glad to see that not only we can achieve inducible nitrogen fixation, we can also achieve ammonium excretion. And so the more we put arabinose, the more ammonium we release. Here we have actually a drop, which is due to the fact that the bacteria not only are responding to arabinose, they also use Klebsiella can also use arabinose as a carbon substrate. So we can achieve inducible ammonium excretion. All right, the second aspect I want to talk about is trying to understand bacterial interaction in the rhizosphere between our diazotrophs and bacteria in the rhizosphere. And for that, we turn to synthetic communities. And the work is a collaboration with Dr. Ophelia Venturelli here in Wisconsin, and Claire Palmer is a postdoc working between our two labs. And what we do is that we developed a 12-member community that represents the rhizosphere of corn, and in which we added, in which we have some diazotrophs. I'm going to show you what that community is in a minute. But what we're measuring in that community is, well, what we do when we do SynComs, we measure abundance of these 12 members over time. So for that, we assess community composition at different time points using 16S sequencing. We monitor nitrogenase activity using the acetylene reduction assay and ammonium production. And with all that, with information that we take, of course, on the entire community that we call PINCOM1 for the first generation. We evaluated that for all the monocultures, all the possible pairs between these 12 members, which means a lot of combinations, and also what we call the leave one out community, where we just remove one member of the community. And with that that information, we build a model, computational model for the community, and that model guides us for acquiring even more data on that community. For instance, the model tells us, okay, now you need to test these 5 members and or 6 members for all these parameters again to refine the model. And so that's really where we are now, and we hope to finish that within literally a few weeks, and to publish that this summer is a model of all the bacterial interaction in the rhizosphere between diazotrophs and non-diazotrophs in the rhizosphere of corn. Here is the community. So we started our base community is based on the work of Roberto Coulter in Roberto Coulter's lab that they published that 7-member community in New et al. In 2017. Several other colleagues I know work on that community as a model community for the corn rhizosphere. And to that community that contains 1 diazotroph, we also added 5 what we call tractable diazotrophs that we are working on in the lab, including, of course, my favorite Klebsiella varicola, but also Pseudomonas suderi, Haber-Bosch, Rhizosphere, and Azotobacter. And to show you the kind of data that we're acquiring, as I said, we are very interested in nitrogen fixation. So I'm just going to show you today the nitrogenase activity, some of the nitrogenase activity data. So here, for instance, that graph shows the y-axis shows the nitrogenase activity of these different combinations of bacteria. The whole community, PICOM1, the level is here. And if you're looking at the activity of the diazotrophs alone, or the 6 diazotrophs alone, the level is here. And so what that means is that the other 6 members, non-diazotrophic 6 members of that community, decrease the overall activity and nitrogen activity of the diazotrophs, meaning overall these 6 other bacteria are competitors. But it's not only a matter of doing that overall, we have all the information, again, on all the pairwise combinations and all the leave one outs to build the model. Here, that's, again, my favorite Klebsiella varicola. That's the level of nitrogenase activity alone. And if you are looking at, that Klebsiella varicola and the one that we call OP, we have a decrease in activity. So OP is one of the many competitors of Klebsiella varicola in the rhizosphere. But interestingly, if you look at KV, Klebsiella varicola, plus the one that we call CP, which is here, is a Cortobacterium, Klebsiella varicola and Cortobacterium, we have an increase in nitrogenase activity. Overall, the pattern that we see is we see a lot of competition and a little bit of synergy. The competition is interesting. The synergy is even more interesting in the short term because we can imagine taking these, what we call helper bacteria of the diazotrophs and use them right away, even as a product, as co-inoculant for co-inoculation to boost the activity of our nitrogen fixers. So we were interested in trying to see, okay, how can we get even more of these helper bacteria for the diazotrophs? And so for that, Dr. Paulo Fernandez, who works for Embrapa, Paulo did a sabbatical in my lab for a year and a half, and Paulo had a fantastic idea. So the idea is that for many of these diazotrophs, we use nitrogen-free semi-solid medium to isolate them. In particular, it works great for Rhizospirillum, Haber-Bosch, and in these media, these nitrogen fixers form a pellicle. And when we take these pellicles, typically, traditionally, I would say to isolate diazotrophs, we often isolate non-diazotrophs, but most of the time we discard. And so Paulo got the idea of what if within these pellicles we can find helpers? And the answer is yes, we can find helper bacteria that help the diazotrophs that are here in that pellicle fix nitrogen in a medium that has absolutely no nitrogen. And that's the kind of data we got, is that we tested here if they help Klebsiella varicola again. And the level 0 means that there is no change compared to the level of nitrogenase activity of Klebsiella varicola alone. And as you can see, well, many competitors, but we actually found that it's a very efficient strategy to identify helpers. And here, that's that helper of Klebsiella varicola in green. It's actually Rhizospirillum brasiliense. But we isolated many other helpers, like that one here in blue, that enhance the nitrogenase activity of Klebsiella varicola. And so for that, we now are very excited about different aspects. One is the practical. We can just take these bacteria as co-inoculants of our engineered strains, but we can also want to understand the mechanisms and how these helper bacteria help our engineered diazotrophs. So we are looking at both the fundamental and practical aspects of that. The last aspect I want to talk about during my seminar is getting some help, not from bacteria, but from fungi. And we turn to arbuscular mycorrhizal fungi. So in my lab, we have several other projects on arbuscular mycorrhizae. And as you probably know, these arbuscular mycorrhizal fungi really help the plant for the acquisition of water, but also nutrients. Phosphorus was very well studied, but also inorganic nitrogen. And so we were wondering, are arbuscular mycorrhizal fungi helping in the transfer of nitrogen released by the diazotrophs, the engineered diazotrophs, and transferring that to the plant? So that's what we have tested. And the work was initiated by Dr. Devanshi Kokani when she was a postdoc in my lab. Devanshi is now an assistant professor at the University of Minnesota, and we continue to collaborate on these tripartite interaction between mycorrhizal fungi, nitrogen-fixing bacteria, and plants. And so what we did with Devanshi, and Devanshi was involved with the work with John Peters on Azotobacter, is that we took our non-fixing mutant of Azotobacter vinlandii. We took one of our really good ammonium excreters that I talked about at the beginning of my talk. And we did experiments called 15N dilution assay, where we enrich the pot with a 15N enriched nitrogen fertilizer, and the plants get their nitrogen from the soil. Their nitrogen content reflects the nitrogen level of the fertilizer. But if you have fixation, nitrogen fixation, there is very little 15N in the air. And so when you have a plant that gets some of its nitrogen from the atmosphere, it's going to decrease the 15N content of the plant. So what we're looking at for evaluate nitrogen fixation here in these assays is we're looking at a decrease in 15N content. So first, when we do that experiment using the non-fixing mutant and the ammonium excreter on corn in the greenhouse and do a 15N dilution assay in red, these are basically different time points where we measure 15N content at 10 days after inoculation with the bacteria, 20 and 30 days. First, we were very disappointed these greenhouse experiments on corn when they were not mycorrhized, that we see no difference between the non-fixing strain and the ammonium excreter, indicating the corn is not getting any nitrogen from the engineered diazotrophs in that experiment. That's until we thought about, okay, what if we put arbuscular mycorrhizal fungi? And when we put arbuscular mycorrhizal fungi, ta-da, that's what you observe. Red is the non-fixer, blue is the engineered diazotroph. And right away, even after 10 days after inoculation of mycorrhized plants, we see a strong decrease in 15N content, indicative of nitrogen coming from the atmosphere, diluting the 15N, and decreasing the 15N content of these mycorrhized plants inoculated with engineered diazotrophs. So the conclusion here is that clearly arbuscular mycorrhizal fungi help in transferring nitrogen from engineered nitrogen-fixing bacteria to the plant. All right, so I'd like to finish just to show you that graph, that figure that I showed you before, of the 4 different strategies to how that people are following to try to improve fixation, biological nitrogen fixation in association with cereals, going from the highly plant engineering approach to what I talk about today, which is engineering the bacteria. And I wanted to address a question that I get most of the time during seminars is, are we there yet? So that was actually the title of a review we wrote in 2019. And well, for the approaches of engineering nitrogen fixation just in the plant or engineering root nodules in biosis, in non-legumes, which is something that I'm working on in my lab, these are, I think, really great approaches, but really long-term. The timeline for the engineered bacteria is now. I'm sure some of you have heard about the work of Pivot Bio or Switch Bio as companies that are already working on commercializing some of these engineered nitrogen-fixing bacteria. I think there is room for improvement, and I think that still for the next 10 or maybe 20 years, there's going to be improvements on the approach of engineering the microbe. I think that engineering the microbe is going to reach a limit also in terms of efficiency of the system, and that we also need to think about engineering the plant, not only better microbes, but also better host plants. And I do think that there is a lot of promise in looking at accessions, so again, of cereals, maize, and sorghum that associate more efficiently with nitrogen-fixing bacteria than our current cultivars. But it's plant genetics, takes obviously more time. So for instance, for my lab, we have initiated the breeding of maize and sorghum, where we introduced that trait of aerial roots into cultivars adapted to the US and to the Midwest, but it takes time. For the corn, we are still doing, it's called the pre-breeding. We hope to be done with the pre-breeding in 4 or 5 years, 4 years, I would say, if everything works well, but it takes time to develop new cultivars. All right, so I'd like to acknowledge people who crossed the funding and people who worked on the different aspects of what I talked about. Funding for me comes from the DOE and the bioenergy, the Great Lakes Bioenergy Research Center, in particular for my work on sorghum. USDA funds my work on maize, and NSF also the bacterial work. And I need to acknowledge funding from the private sector, from the Pivot Bio company. And of course, I need to acknowledge the fantastic work of my lab members, in particular for the work I presented today, Valentina Infante, Bijwajit Samal, Junko Maeda, and April McIntyre. April works now for actually Valent Biosciences. Also I want to acknowledge collaborators on these projects. Brian Fleger here at the University of Wisconsin is a fantastic collaborator for synthetic biology. Ophelia Venturelli is our collaborator for the work on synthetic communities. Paulo Fernandez from Embrapa, who did a sabbatical in my lab for several years, is still a great collaborator. Devanshi Kokani is now at the University of Minnesota, and John Peters from the University of Oklahoma. Thank you very much for your attention. I will be glad to answer your questions.
Thank you very much, Jean-Michel. That was really a great presentation and really went into a lot of detail about the different statuses. We do have a number of questions related to the transfer from the greenhouse to the field. What do you think the main cause of that is? Is it the environment in which it is? Is it the biome? Is it weather? What are you thinking that may be the reason?
Yes, to all. Yes, yes. The environment, including the microbial environment, are different, very different. The soil, the texture, structure, it's the environment. It's clearly the effect on the environment. I can say that our best ammonium excreters in the greenhouse were wiped out. Like, we're not competing at all in the field.
Wow.
And so everything we did in the greenhouse was more or less useless. And so I really think that we need to evaluate nitrogen fixation as quickly as possible in the field. It's not easy. That's something about nitrogen fixation in general. For cereals, we have 5 categories of techniques to evaluate nitrogen fixation. And all of them have pitfalls. For the field, the one that I think is the ones that are the most reliable are 15N dilution and 15N and nitrogen balance. But these techniques it's low throughput, and it's expensive, and yeah, evaluating nitrogen fixation in the field is possible, but it's a lot of work. And I need to say that without the funding of private companies, it would have been sometimes difficult for me to do that, or the scale would have been impossible. Yeah.
Yeah, so I wonder, have any research actually been done on using these in controlled environment agriculture? I know we're not talking maize and sorghum at that point. We're talking, more of the produce species. So do you know of anything?
Yep. No, I've not. For me, I really focused for cereals on maize and sorghum. So that's
Right.
where I think there is clearly a very interesting potential for other crops. And again, I think it should be done in as quickly as possible in field conditions, because
Yeah, so we have, at least from what we always hear, it's field first. Get in the field, because when it deals with biology, you really have to look at it from that standpoint. So let me get to one of the next questions. Is there any plant-derived signal that can also be used as a helper for enhanced nitrogen fixation? Have you explored that possibility?
I know that there are some companies that think they have products that do that. I don't have a personal experience with that. It's possible. I think it's something to explore, both on the practical side, but also getting a mechanistic understanding on how these molecules work. I think it's also would help to solidify the claims. But sure, it's absolutely, I think, worth pursuing. Yeah.
So you mentioned checking the results of nitrogen-fixing bacteria in the field. How do you think this might affect the nitrogen-fixing metabolic pathways of the rhizosphere?
It's a great question, and something that I think is actually not explored enough. Sure, it's obvious that even in our SynCom experiments, when we start putting an ammonium excreter, it does disrupt the bacterial interactions. All the bacteria love ammonium. Many of them are the nitrogen which is released by the engineered strains is clearly taken up to some extent by other bacteria in the rhizosphere, without any doubt. And so for sure, it's gonna change the rhizosphere composition. Yeah.
So when you're picking your synthetic communities, how do you do that? How and why are you picking certain ones?
It's endless discussions on how we do that. So it's complicated. So you have different ways of picking them. One is, by taxonomy, which is the easiest one, maybe the worst one, because of course we want to pick them also by function. So for us, what we do is that before making a decision to add a new microbe to the community, we do a whole genome sequencing. We try to do metabolic modeling to try to predict its functions as much as we can. Looking at transporters of these bacteria is often a good way to have an idea of what they like to eat. So as much as it's, for us at least at this point, it's a combination of taxonomy and function.
Right. So how advanced do you think we are in terms of function, understanding function? I knew you would do that.
Not far enough. Yeah, obviously not far enough. I think it's really interesting to, yeah, absolutely. We need to I think the approach, for instance, that Carsten Zengler at UC San Diego is following, a combination of where he's looking at transcription efficiency of these different bacteria, trying to group bacteria into functional guilds of bacteria that perform similar functions in the rhizosphere. I think it's very interesting. And I think clearly that's where we need to go in trying to understand all the biome system as a whole, and trying to group bacteria into bacteria that have similar functions is absolutely necessary. Again, I think the work of Carsten is really enlightening, that when he removes one of the members of the guild, he sees all the other bacteria of the same guild increasing in abundance, indicating that within the guild you have competition, and these different group of bacteria performing similar functions interact themselves. So I think that's how we need to think about the system over now, as much as possible, not of course in taxonomy, but a lot in function. Yeah.
Yeah. So you know, you mentioned that Klebsiella was your favorite.
Yep.
How does that remain? How long does it remain stable or effective throughout one farming season, for example?
Yeah. So for that, I would rely on data, I mean, from Pivot Bio, and where basically Klebsiella, at least the engineered strains, are not detectable at the end of the season anymore in the field. What I can say in terms of abundance during the season is that they clearly spike after a few months in the field in the corn rhizosphere, and then there is a decrease around testing time. So clearly, these bacteria, Klebsiella and others that we work on, are strongly dependent on plant carbon. So I know that we probably, and actually literally in our recent review, we need to think more about biocontrol of these strains. There are some concerns, and I think people could make big mistakes by releasing, for instance, free-living bacteria, free-living diazotropes in the field. For instance, I have my Azotobacter. I mean, we have our Azotobacter, which is a great salt bacterium, but we have very little control over Azotobacter because it can take a wide range of carbon sources, and would remain in the field for quite a long time. So I never put Azotobacter, even if legally I could, these engineered Azotobacter in the field, because of concerns of biocontrol. The strains that we currently put in the field are all strongly dependent on root exudates. Without corn root exudates, these bacteria basically are undetectable. So that's our form of biocontrol right now, working on associative diazotrophs and not free-living diazotrophs. But I think we need to think about even more stronger ways to have biocontrol over these strains. The problem is many of the techniques we currently use for biocontrol are based on transgenic approaches, and that raises all that.
It raises just different questions that come in, and certainly from the regulatory perspective as well.
Yeah.
So, you know, generally in what at least from a regulatory standpoint, they're looking at, you know, does it dissipate? And if you can't control it, then there are serious questions about whether you can actually get a permit to release it. So
I can say that Klebsiella is strongly dependent on plant carbon, which provides some level of biocontrol. The fact that we're also working with engineered strains where their fitness is actually not increasing but decreasing provides some level of biocontrol too.
Yeah. But again, that makes it extremely hard from a regulatory perspective to be able to show that consistently in field environments, I think. But So I have one last question. We have a whole lot of other questions, and just so that everyone knows, we hope Jean-Michel will answer those questions in writing or reach out to you directly, and we will put them on the YouTube channel so you can actually see that. So if you had your wish for the next 5 years of research, not just in your lab, but generally in this area, what would you like to see? If you could, let's say you've got $500 million or that you could actually spend to accelerate the research and our understanding of the functional aspects and the interactions between them. What would you see as the main research priorities?
One would be more field experiments and quantification of nitrogen fixation, careful quantification of nitrogen fixation across different environments. The second one would be more research on the plant side, on hosts, because I feel that we are soon going to reach a limit of what we can achieve by just engineering the bacteria. And so I think that we need better crop cultivars, and not only in maize and sorghum, and we need plants that would provide a better environment for these bacteria to get carbon and a niche to fix nitrogen. So I think even if it's more difficult and requires more time, I think we need better plants, better crops and in terms of hosting these bacteria without impacting the yield, which is the difficulty. We want to have
That's correct.
That would provide carbon. They will have to provide carbon for these bacteria, but without significantly impacting the yield. So, yeah.
Though very little is going on in that at this point in that area. I mean, at least from our approach within the Phytobiomes Alliance, we're really trying to encourage that cross talk, because we really want the different disciplines to start thinking about how we do this together, and how we can take the bacteria and the plant side and then really try to create ideal systems for particular environments. And that's challenging. So
Yeah,
I appreciate your thoughts on that. Again, a fabulous webinar, and I appreciate your sharing your thoughts with us today. And we look forward to seeing you. And I hope everyone, or as many as possible, will join us in St. Louis later this year at the Phytobiomes Conference. So thank you for participating today and joining us. Look forward to seeing you in person.